Good evening, everyone. So this week we'll be continuing our discussion on connective tissue disorders. Last lecture was on anti-nuclear antibodies, and I hope that it had made the understanding a bit easier. Okay. Now, after we have had a background of anti-nuclear antibodies, we can continue our discussion on connective tissue disorders. The pathophysiology of SLE is actually very complex. It's in my opinion the most complex topic that I have tackled in the last more than 120 videos. So what I've done is I've decided to make it simpler to understand and easier to understand with important concepts that you can use to have a better understanding of SLE, to understand the background of SLE, the pathomechanism of SLE. So if you have a good grasp of the concept behind why does SLE develop, or what are the various processes that leads to the clinical outcome that is known as SLE, so we can have a better understanding of what are therapeutic modalities we can use to treat SLE. Okay. Now, this video lecture will just discuss what is happening at the level of cells, at the level of molecules, and how does the body actually develop SLE. Okay, so without much further discussion, we shall start our lecture on pathophysiology of SLE. Let's begin. Now, SLE is a systemic disease characterized by multi-system organ inflammation. Now, this term is very important. It is multi-system organ inflammation. That means inflammatory damage to end organs involving multiple systems. Now, most the symptoms include so the system includes skin, joints, and vasculature, where the end organ damage and vessel damage are the hallmark. So there is, vasculitis and vessel inflammation, and that is quite important. Okay. Now, SLE exists because of an complex interplay between the host factors like the genetic makeup of the individual and the hormonal milieu, etc., along with environmental factors like UV radiation and all the triggers, infections, viruses, bacterial infection, drug intake. So all of these triggers from the exogenous milieu or the environment, plus in a genetically susceptible individual, causes SLE. Essentially, SLE is characterized by loss of self-tolerance, which leads to induction of autoimmunity, which leads to activation and expansion of the immune system, which leads to immunological injury to end organ, and clinically we have SLE. So what happens is we know that our immune cells don't react or don't surmount an immune response to our cells, to host cells. That is because they have something which is known as tolerance. The immune cells are tolerant to the self antigens, and that is why the immune system is able to function normally. But in SLE, this tolerance is gone away. Okay, this tolerance is subdued, it is decreased, and because of the decreased tolerance, the immune system starts to recognize normal cells as foreign cells and start to produce antibodies. In the case of SLE, the target antigens are DNA, RNA, nuclear material, components of the cell membrane, cytoplasm. That's why you have all those different antibodies like anti-dsDNA, anti-ssDNA, anti-RO-LA, anti-RNA, antiphospholipids. So these kind of antibodies occur to self-antigens. When the self-antigen antibodies are presented and then they form complexes with the antigens, these immune complexes get deposited into end organs and start damaging them. When that damage occurs clinically, the patient is known to have SLE. Clear? Let's move forward. The male to female ratio is about 7:15 to as high as 1:9. That means 9 times more the female are more susceptible to develop SLE. And we know that fact that autoimmune disorders are actually more common in the female patients. Okay. And this data is actually very important to remember, that you can quote in your exams or in your vivas. It's about ranging from 7:15, that is twice the amount of males, and it can be as high as 9 times the amount of males. However, males have higher frequency of renal disease, skin manifestations, cytopenias, serositis, CNS involvement, thrombosis, and vasculitis. So they have higher frequencies of these disorders. Age is roughly around 38 years in females and about 44.2 years in male. So it's a issue of the fourth to fifth decade. In case of pediatric SLE, it's uncommon, but roughly about 20% of all SLE can be pediatric cases. In the case of pediatric SLE, the onset is this earlier onset of organ damage, and there is higher disease activity at the onset. That means pediatric SLE, about 20% of all SLE cases, higher organ damage at the onset of disease, and earlier onset of organ damage is seen in pediatric SLE. So, before we start discussing the pathomechanism of SLE, what I've done is I've divided into certain parts, okay? So that if we can discuss individual aspects separately, it will be easier for us to understand. But remember, everything is working together and in connection with other aspects also. So, an individual will have a genetic basis of SLE, a genetic component, some environment trigger. Most common trigger or the most important trigger to understand is UV radiation. Innate immunity activation via toll-like receptor 7, interferon 1 or NETs. We'll discuss each of them separately subsequently. Or adaptive immunity activation, that means T cells or B cells which have been sensitized in an LE individual against autoantigens, decreased T regulatory cells, and the development of autoantibodies. Autoimmune antibodies, and we'll discuss very shortly about dsDNA and all the antibodies, but we go in much detail in our last week's lecture on anti-nuclear antibodies. And then all ending in end organ damage. So all of these factors, they lead to end organ damage, and that is what clinically we term it as systemic lupus erythematosus. Clear? So let's discuss each of these aspects separately. The first aspect is genetic basis of SLE. Now, in genetic basis, one thing to understand is that the main pathway responsible is interferon 1. Now, this interferon 1 or the type 1 interferons, which includes interferon alpha, beta, I think delta and kappa. So, all those interferons are part of family, a larger family of interferons known as interferon type 1. Okay. So, an individual who develops SLE has a genetic predisposition to secrete interferon 1 cytokines. Okay, so you have an individual and the genetic makeup is so that the immune cells of the individual are more prime, more susceptible, more ready to secrete type 1 interferons. When type 1 interferons are secreted, you have generation of pathogenic autoantibodies. So these interferon type 1 interferons along with pathogenic autoantibodies leads to development of the systemic lupus erythematosus like features in an individual. Clear? So the genetic makeup that decides how your immune system will react to antibodies, if that is not properly created because of any kind of genetic mutation, you can have development of autoantibodies. And when autoantibodies start to develop, these autoantibodies will react with autoantigens or the antigens in the body, form immune complexes, and then lead to end organ damage. In fact, it has been found in murine studies that genetically identical mice, about 25% develop SLE. That means 1/4 of the mice population with mutations developed SLE. That means mutation is not the only reason. You have all the other factors working to develop SLE. And that's what we discussed in last slide, that we had at least 5-6 different factors that actually come together to lead to development of SLE. One HLA that has been implicated in SLE is HLA-B8DR3, which has been found as an individual risk factor for SLE. In fact, individuals with HLA-B8DR3 positivity have shown to have lower values of interleukin-2. And actually interleukin-2 is a very important cytokine for T regulatory functions. When interleukin-2 is decreased, the T regulatory function doesn't happen properly. And the function of T regulatory cells is to regulate any immune reaction, whether it is because of any infection or whether it is because of any immunity. Okay, so if interleukin-2 is decreased, the two T regulatory cells are not able to function properly and they are not able to regulate immune reaction. So if any kind of autoimmune reaction occurs, it is not regulated and then it can lead to SLE. Clear? In fact, we see decreased IL-2 levels in patients with SLE. Another mutation like early complement gene mutation C1q has been found in SLE patient, and that leads to increased anti-dsDNA antibodies, a severe disease, and childhood onset of SLE. So if you have mutations like mutation in the C1q complement pathway, in the complement pathway we have many different proteins. We already have a separate lecture on complement pathway. You can search for it and look at it. So if there's a mutation in the C1q, you have increased anti-dsDNA antibodies, a severe disease, and an earlier onset of disease. The second aspect that we'll discuss is environmental triggers, namely UV light, drugs, viruses like herpes virus, SARS-CoV-2, Epstein-Barr virus is one of the important viruses. It induces mitochondrial stress. In fact, EBV DNA has been found to initiate production of interferon type 1 by the plasma cytodendritic cell and also to increase proliferation of B cells. Clear? So EBV DNA, Epstein-Barr virus DNA, has been distinctly studied in the pathomechanism of SLE. Other viruses like herpes and COVID has also been implicated. In fact, gut microbiome, Enterococcus gallinarum, is also being studied as a potential implicant in the SLE pathomechanism. Now, all of these environmental triggers, they increase DNA damage. So what do we mean by that? If you have a cell, let's say keratinocyte in the skin, and it gets damaged by UV radiation, the cell will die. Isn't it? It's getting damaged, so it will undergo apoptosis. Whenever a cell dies, it will release all the cellular debris or the cellular remnants. And these cellular remnants, if they are not cleared properly by the immune system, they will get exposed to the immunity, and then they will develop autoantibodies. And if the autoantibodies are against nuclear antigen, they will be called as anti-nuclear antibodies. Nuclear antigens or the cell structure antigens, cellular organelle antigens. And this ANA, when they develop, it leads to SLE and other connective tissue disorders. Clear? Let's move forward.
Hi. Hope you're enjoying this video. I know it's a long video, so I thought I'll give a certain break after listening to such a long videos that I make. If you think that these videos are helpful in your residency, do consider becoming a member. You can opt for Sapphire membership or Ruby membership. I would prefer it to be a Sapphire membership, of course. You get access to longer videos, bonus contents, extra information to make things more interesting, and to cover each and every aspect of the topic that we are discussing. Okay, so I think that's enough of a break. I should continue with the video, and you should listen to the other person at the bottom right corner tell you different topics of dermatology, venereology, and nephrology. That being further said, let's continue with the video.
So let's discuss UV radiation in detail. UV radiation is one of the most important environmental factor in the induction phase of SLE, a phase from which the SLE starts. Now, UV radiation leads to apoptosis of keratinocytes, and when the cell undergoes apoptosis, there will be leakage of nuclear and cytoplasmic component in the circulation. When these components go into circulation, they can induce autoantibody formation. Okay, so normally if a cell is destroyed or dies away, the components which are released in circulation are rapidly cleared up by macrophages, T cells, and B cells. Now, these macrophages, T cells, and B cells, and all the immune cells have tolerance. So they know that these antigens are the body's antigens. So they don't make antibodies for it. But if the response is not proper, if the immune system is genetically susceptible to form autoantibodies, the macrophages, T cells, B cells, NK cells, the immune cells, they will recognize this cellular material and form autoantibodies. Okay. So when there is a loss of tolerance and self-immunity, you have development of autoantibodies. And when autoantibodies are formed, you have SLE and connective tissue disorders. Now remember, these diseases are known as connective tissue disorders. That is because they present as joint pain and all muscle involvement and all. But actually, these are inflammatory disorders. They are disorders of inflammation, disorders of decreased tolerance in the immune system. So they are immune-mediated disorders, autoimmune disorders. So that's a better term to use rather than using CTDs. Let's move forward. The third aspect to discuss is the innate immunity activation. Okay, so we have discussed the role of environment in the form of environmental triggers, UV radiation being the most common one, and infection by any other viruses that can stimulate the immune system in a wrong way so that there is development of auto-antibodies. Clear? Now we'll discuss what happens at the level of innate immunity, at the level of the NK cells, T cells, and all the antigen presenting cells, the dendritic cells. Okay. Now what happens is, and I would request you to go back and see our lecture on basic skin immunology, innate immunity, to have a better understanding what are the players involved in the innate immune system. So let's start what actually happens. Okay. The most important cell that we'll discuss are the plasmacytoid dendritic cells. Okay, plasmacytoid dendritic cells. Now, these dendritic cells are responsible for analyzing foreign DNA and RNA matter. So, if any kind of bacteria, virus enters the body and leads to release of any nuclear material like DNA or RNA, they are recognized by plasmacytoid dendritic cells. Now, these dendritic cells, after recognition, will secrete interferon type 1. Now, this type 1 signaling will eventually lead to secretion of interferons like interferon alpha and beta. Okay. Now, what happens is that this is the normal pathway, the normal function of plasmacytoid dendritic cells. If this functioning, instead of recognizing foreign DNA or RNA, starts to recognize self, then the immune response will be against the self antigens. And instead of immunity, you will have autoimmunity. Clear? Now, interferon 1 or the interferon type 1 is the most upregulated gene pathway in SLE. So remember, for example, in psoriasis, the main pathway is TNF alpha. For your vitiligo, the main pathway is interferon gamma. In this case, it is interferon type 1 group of interferons. So it's the most upregulated gene pathway in SLE and is increased in monocytes and keratinocytes more than the lymphocytes. Now, this stimulation of secretion of type 1 interferons, that means you have a plasmacytoid dendritic cells here, and this will lead to formation of type 1 interferons. This secretion of type 1 interferons is mediated by the activation of toll-like receptors 7 and 9. Basically, two toll-like receptors 7 and 9. And by the action of TLR7 and TLR9, it leads to secretion of type 1 group of interferons. Clear? We'll discuss each of these steps in details in subsequent slides. Let's move forward. Now what happens is that if you have any immune complex formation between the autoantibodies and self DNA and RNA from destroyed cells, now these can be recognized by plasmacytoid dendritic cells. Now what plasmacytoid dendritic cells are responsible? They are responsible to recognize DNA and RNA from the outside of the body normally. But in the case of autoimmunity, they start to recognize self-DNA and RNA material from destroyed cells. If there is overactivation of plasmacytoid dendritic cells, it can increase interferon 1 stimulation or interferon type 1 stimulation. If this stimulation is long-standing or the chronic type 1 stimulation can lead to stimulation of B and T cells. And when B and T cells are stimulated, they will get converted to cell population known as lupus T cells and lupus B cells. This is just a term to show that these are not normal T cells or B cells. They are the cells of the individual with lupus, and they don't have that robust self-tolerance. Their self-tolerance level is decreased. So they can recognize autoantigens and form autoantibodies. Clear? So what happens is plasmacytoid dendritic cells recognize self-antigens, and after recognizing, they can stimulate TLR7, TLR9 and secrete interferon type 1 interferons. When these are chronically secreting interferon 1, the stimulation can stimulate B and T cells, convert them into lupus T cells and lupus B cells, which cells have decreased tolerance, and because of this decreased tolerance, they lead to inflammation and immunity. T cell leads to cytokines, inflammatory cytokines. B cells leads to autoantibodies, and together they lead to connective tissue disorders or autoimmune disorders like SLE. Clear? Let's move forward. The role of toll-like receptor 7 is very important in SLE. In fact, toll-like receptors, when they are when they are stimulated by signaling molecules, they lead to activation of number one, the interferon regulatory factor family, the NF-kappa B family, and the mitogen activated protein kinase family. Now, all of these families are required for proper inflammatory cascade. Okay, we know that NFK beta is a very important mediator of inflammation. We have discussed that when we were discussing drugs like cyclosporin or the pathomechanism of vitiligo and psoriasis. We have discussed NFK beta. Now, what NFK beta does is NFK beta goes inside the cell, inside the nucleus where you have the DNA, and this NFK beta, when it attaches to the NFK beta response element, this leads to secretion of inflammatory cytokines. So what we are saying that plasma cytodendritic cells activate toll-like receptor 7 and 9, and after activation they increase the NFK beta mediated inflammatory cytokine secretion. Also, the interferon type 1 factor family is also increased, so you have more secretion of interferon alpha and interferon beta and kappa, delta and all that. The MAP kinase pathway also increase the production of inflammatory cytokines. Now, what actually is happening? The plasma cytodendritic cells are responsible for secretion of interferon 1 and also secretion of all the cytokines required for inflammatory pathway, and all of this comes together to increase the inflammation inside the body, leading to SLE. Now, look at this diagram. This is a diagram of plasma cytoid dendritic cells. So it shows all the methods that which toll-like receptors can get activated. You have the mitochondrial DNA, and you will ask why is mitochondria outside the cell? Because this mitochondria has come from other cells which have been destroyed. Okay, so all these cells undergoing apoptosis, they release their debris in the circulation, which can be recognized by plasma cytoid dendritic cells. These cells can also recognize anti-DNA antibodies secreted by in a patient of LE, apoptotic cell debris, Epstein-Barr virus. Remember, they are suited to recognize RNA and DNA. EBV encoded small RNAs or any RNA attached to the anti-RNA binding protein antibodies. So any of this stimulation can attach to the receptor, which is the FC receptor, and after attachment, they can stimulate and activate toll-like receptors 7, 8, and 9. When this happens, there's a cascade of all the inflammatory molecules which can increase the production of interferons or the inflammatory cytokines and all the interferons, namely interferon alpha. So you can see the NF-kappa B going inside the nucleus, or IRF7. IRF7 is, I think, interferon response factor 7. It goes inside and it increases the translation, increases the transcription of all the inflammatory cytokines and interferons. Okay, let's move forward. Hi everyone. Hope you're liking this lecture, and I hope you're finding studying HIV, beneficial. Again, I'm coming you to request again for the bid for Delhi for Dermacon 2028, and from support of IDBL Delhi branch, I would like to request you to bid for or to support us in organizing Dermacon 2028. So let's come together, let's meet in Delhi and take part in this academic extravaganza known as Dermacon 2028. Let's see you in a few years from now, and happy learning. Now let's discuss nuclear extracellular traps, or NETs. NETs is a very important concept to understand in the pathomechanism of LE. Okay. Now what happens is that there is interaction of neutrophils, which are the acute inflammatory cells. Okay, they are the first cells to arrive in the case of inflammation. Acute inflammatory cells, when they interact with vascular endothelial cells or any activated platelets or any inflammatory cytokine, they lead to formation of NETs, or the nuclear extracellular traps. So what are NETs? NETs are aggregates of nuclear material and proteins excluded out of neutrophils. So let me just explain it very easily. You have a neutrophil, and this is a bilobed or multi-lobed nucleus. This nucleus or this neutrophil, when the neutrophil want to stimulate the immune system further, they can extrude or push out material of the nucleus outside the cell. When this happens, it will get broken up. So this will lead to all the broken up formation. So all the molecules like mitochondria derived DNA, histones, HMG1, LL37, which is one of the antimicrobial peptides, myeloperoxidase, nucleosomes, RNA molecule, all this cellular debris, the other immune cells will recognize it. And when they recognize it, they will think something is happening. Neutrophils is trying to call us. Let us increase the inflammation and help neutrophils. And that is how neutrophils, by the production of nuclear extracellular traps, increase inflammation. So why they are known as nets? Because they come from the nucleus, which are extracellular, and this is along with all the protein molecules, all the other molecules are trapped together to form nets. And these nets act as stimulation for the immunity, stimulation for the inflammatory process. And when this inflammatory process goes unchecked, you have autoimmune disorders. Clear? Let's move forward. So what happens when nets are produced? These nets can act as source of relevant self-antigens. Remember, I've told you how nets are constructed. They are constructed by broken down nuclear fragments along with all the protein molecules. If you have broken nuclear fragments in the circulation outside the cell, they can stimulate immune cells to form autoantibodies. Okay. Normally, the genetic material is safe inside the nucleus. If it goes outside the cell, it is prone to get recognized by immune cells and lead to formation of autoantibodies. Now, these net can individually induce interferon type 1 from plasma cytodendritic cells, thereby increases inflammation and the interferonopathies or increased type 1 interferons. Now, these nets can also cause vascular damage. Remember, they are secreted because of interaction of neutrophils with vascular endothelial cells. They can cause vascular damage because inflammation can also lead to thrombosis. The process of formation of nuclear extracellular traps is known as netosis. Clear? Let's move forward. The fourth aspect that we will discuss is the adaptive immune activation. So till now we have discussed genetic makeup of the individual, environmental triggers, the activation and overstimulation of innate immunity. Now we'll see the adaptive immunity. Now in adaptive immunity, we'll first discuss the T cell function. T cell in lupus, normal T cells, they are not a normal T cell, it's T cell in the patient with lupus. They provide helper signals and cytokines to differentiate B cells into autoantibody producing cells. Okay, so what happens is that T cells can stimulate B cells in the setting of lupus to secrete more autoantibodies. Okay, along with that, lupus T cells have been shown to have activated mTOR. That is why all the mTOR inhibition is one of the therapeutic modality. They can have altered mitochondrial oxidative pathways. Remember, oxidized mitochondrial mitochondrial DNA, mitochondrial genetic material can lead to activation of inflammation and interferon beta through the toll-like receptor independent pathway. These T cells can show CD40 ligand, and we remember that plasmacytoid dendritic cells have CD40 receptors, and when the ligand gets attached to receptors, you can have stimulation and formation of interferon type 1. These T cells show decreased production of interleukin 2, and because we have told you that interleukin 2 is important for T regulatory function, IL-2 is decreased, T regulation is decreased. If T regulatory cells are like class monitor, if they are decreased, they are not able to take care of the inflammation, and the inflammatory pathway goes unhindered and unstoppable. Clear? And these T cells in lupus can increase the cytokines for B cell differentiation like IL-21, etc. So they can overstimulate B cells or stimulate B cells in such a way that they start secreting autoantibodies. Clear? Let's move forward. So what happens when B cells are activated? Now what happens that these B cells, when they're activated, they can also act as antigen presenting cell to lupus T cells, further increasing stimulation. Now remember the dogma of immune system. Any immune response will increase itself, which is known as positive feedback. will make efforts to stop or decrease itself, known as negative feedback, and also stimulate other processes. So that is what is happening. The B cells, activated B cells in lupus, are stimulating T cells by acting as antigen presenting cells. These activated lupus B cells are responsible for production of autoantibodies. These B cells have an hyperactivity status. They are working more than usual, and they also can be easily stimulated. So the threshold of stimulation is also lower in B cells in lupus patients. Clear? Let's move forward. So let's discuss the important aspect, the anti-nuclear antibodies in SLE. So anti-nuclear antibodies is positive in about 96 to 99% cases of SLE. So it's the first autoantibody that you check off, you get an ANA done. Now, after ANA is positive, you go forward and look for specific autoantibodies. If you look for anti-dsDNA antibodies, it is associated with glomerular nephritis and correlates well with disease activity. Anti-Smith antibody is highly specific for SLE, but can be positive for other CTDs also, and it is a very important autoantibody to look for. There are cases in which ANA is negative or anti-dsDNA is negative, but anti-Smith is positive. Okay. Anti-Rho and anti-La are important in the case of mothers of children with neonatal lupus and or congenital heart block. So if the mother has anti-Rho or anti-La antibodies, their children during development can have congenital heart block or with or without neonatal lupus. Antihistamine antibody, specific antibody that you look for in drug-induced lupus. But always remember, you have to keep clinical correlation in mind. If the patient is clinically resembling SLE, even if ANA is negative, you can go for specific antibodies. Even if the antibodies may be negative, you can still consider a diagnosis of LE if clinically it fits the gambit of LE. If you want more and a more intellectual, comprehensive, and a deeper understanding, go back to our lecture on anti-nuclear antibodies, and we have discussed each of these antibodies in much detail. Okay, let's move forward. So this is the table that I've created as a summary for all the anti-nuclear antibodies in SLE. We have discussed a similar table. In fact, this is the part of a bigger table that we have discussed in the last lecture of ANAs. So we'll just go through it very quickly. You have anti-dsDNA antibodies against nucleosome or chromatin, anti-Smith against spliceosome proteins, anti-U1 snRNP against spliceosome proteins, anti-Rho against Rho proteins, anti-La against RNA polymerase 3 proteins, anti-ribosomal against the 60S ribosomal unit protein, anti-histone against nucleosome, anti-Q against Q heterodimer. Okay, and these are all the implicated autoantibodies. The group is larger, but these are the major ones of autoantibodies in SLE. In fact, go back to the last lecture on ANAs, and we have a bigger table there, which includes antibodies against target antigens in systemic sclerosis and also in dermatomyositis. Okay, let's move forward. Now, end organ damage in SLE. So what happens at the end, at the clinical damage? All the inflammatory cascade leads to end organ damage. So you have autoantibodies. When they react with target molecules, they form immune complexes. These immune complexes, when they're recognized by immune cells, they activate inflammation, majorly the interferon type 1 interferons, and also complement system activation. These immune complexes can also lead to release of reactive oxygen species and neutrophilic enzymes, majorly through nets and other processes. All of this leads to inflammation and inflammatory damage to the end organs, and after damage, it leads to scarring, leading to dysfunction of the organ. So in these three lines, I have condensed the pathomechanism of LE, what actually is happening. Okay. When T cells and B cells are primed because of this inflammatory cascade, they can lead to persistence of inflammation and relapse of disease. T cells can secrete inflammatory cytokines, B cells can secrete antibodies, and the persistence of antibodies and inflammatory cytokine can lead to persistence of disease. If the damage occurs on vascular endothelium, we can have all sorts of end organ perfusion damage. Okay, in fact, periarteriolar concentric onion skinning spleen is one of the first pathology that was seen in SLE patients. We know that in SLE patient we have microangiopathy, microthrombi, endothelial dysfunction, vessel damage, All of this can lead to premature atherosclerosis and premature sclerosis, leading to end organ damage. In fact, if you remember the biopsy or histopathology of DLE, you will see that there's perivascular inflammation throughout the dermis, isn't it? So throughout the dermis, perivascular lymphocytic infiltrate along with the lichen or tissue reaction pattern is the characteristic feature, histopathological feature in LE. Clear? Let's move forward. Now, finally, these are some reading recommendations. I went through a lot of different kind of articles to have a basic understanding of LE, and I would recommend that you should read the Fitzpatrick chapter on LE first, the basic one. Okay, it's the least that you should do is read. Otherwise, Crow MK's article on pathomechanism of LE and the role of UV radiation by Kuhn et al. Were very good. The This article, Pathogenic Aspects of SLE with Emphasis on Regulatory T cells, is very nicely written article to understand the role of regulatory T cells. Clear? So these are some reading recommendations. But if you have understood this video lecture, that is enough, more than enough that you require to know about LE. With that, again, I will not take much of your time. Request to be a member, but do know that I plan to discontinue to be membership in the coming months. But if you really like these videos, do become a member. And if you think that these videos are important, and if you have somehow gained from looking at these video lectures, then consider becoming a Sapphire member of the channel. It gives you access to longer videos, better content, extra content, and exclusive polls that I put for Sapphire members to decide what newer topics I should tackle. Okay. With that, I would like to finish this week's lecture on pathomechanism of LE. I hope I've made it clearer. I made it easier for you to understand and go forward. If you know what is happening in the background of LE, it becomes understanding the disease better. If you know what is happening at the level of cells and the cytokines, you know that at what level you should use what kind of immunosuppressive agent. And just reading about the pathomechanism of LE is very interesting, if you get my drift. So I would not take much of your time. It's already more than an hour long. If any doubts, just put a timestamp and mention in the comment section. Till then, adios, and I shall see you next week. Bye bye.